Good morning, good afternoon, and good evening to everyone joining us from around the world. My name is Sabrina Welsh, and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for this webinar. On Tuesday this week, a group of 9 drug developers pledged they would not release any COVID-19 vaccines that did not follow rigorous safety and efficacy standards. The pledge was signed by the CEOs of the companies and outlined a united commitment to uphold the integrity of the scientific process as they work toward potential global regulatory filings and approvals of the first COVID-19 vaccines. Later that same day, AstraZeneca announced a clinical hold in their phase 3 trial of a vaccine candidate after a patient experienced an unexpected illness. As Francis Collins of the NIH noted yesterday in a Senate hearing about vaccine development, this is a concrete example of how even a single case of unexpected illness is sufficient to hold a clinical trial in multiple countries, and evidence that we cannot compromise on safety. This example highlights that vaccine development is hard, but it reminds us how absolutely critical it is to go through the necessary testing stages, not only to create a safe and effective vaccine, but also to maintain the trust of consumers who will one day be offered the vaccine. So just a note before we begin, the information presented today includes some pre-published data that is currently under peer review. At the request of the author, we ask that you not reproduce or disseminate the data presented. This session will be recorded and available on our website and our social media channels for you to review. Today's speaker is Dr. John Wherry from the University of Pennsylvania. Dr. Wherry is the Barbara and Richard Schifrin President's Distinguished Professor and Chair of the Department of Systems Pharmacology and Translational Therapeutics in the Perelman School of Medicine, and Director of the UPenn Institute for Immunology. Dr. Wherry has received numerous distinctions and honors, including the Distinguished Alumni Award from the Thomas Jefferson University, the Cancer Research Institute's Frederick W. Alt Award for New Discoveries in Immunology, and the Stand Up to Cancer Philip A. Sharp Award. Dr. Wherry's research has pioneered the field of T-cell exhaustion, the fundamental mechanisms by which T-cell responses are attenuated during chronic infections and cancer. His discoveries helped identify the role of PD-1 and the ability to block this pathway to reinvigorate exhausted T-cells. His group also first demonstrated that targeting multiple co-inhibitory receptors simultaneously could synergistically improve therapeutic efficacy, the foundation for current combination immunotherapy efforts in humans. Dr. Wherry's work has defined the transcriptional and epigenetic atlas of exhausted T cells, defining exhausted T cells as a distinct immune lineage. His laboratory has pioneered the concept of immune health using systems immunology approaches, most recently applying this concept to COVID-19. In today's lab meeting, Dr. Wherry is going to talk about deep immune profiling in SARS-CoV-2 patients and applications to vaccines and therapeutics for COVID-19. During the presentation, please send me your questions using the Q&A function in Zoom. I'll ask our speaker a broad selection of your questions after he's finished the presentation. And please remember to send them in Q&A in the function, not in chat, please. We'll have about 25 minutes for discussion, and I hope you'll take the opportunity to ask your questions about the data presented today. It is now my pleasure to introduce Dr. John Wherry from the University of Pennsylvania.
Well, thanks, Sabrina. Thanks for the very kind introduction, and it's really nice to be here. I see a lot of old friends on the participant list, so it's fun to share some of our new data with this group. Let me go ahead and get the slides up. And hopefully that's all okay, or one of you will yell at me. So what I thought I would do is take you through a little bit of recent data that we published on looking at COVID-19 immune responses in hospitalized patients, how that's helped us understand the heterogeneity of immune responses and define what types of immune responses might be associated with clinical pathology and/or resolution. And then at the end, I'll touch just a little bit on some of our new data from pediatric COVID-19 infection and how those studies are actually, I think, helping us understand at least one potential outcome of adult infection with SARS-CoV-2. This topic really needs very little introduction these days. Still, I put a slide in just to sort of frame some of the questions that we're interested in. We do know a little bit now about COVID-19 immunology, not as much as we would like. We do know that many patients experience an elevated inflammatory response, and that inflammatory response has been associated, at least in some cases, with pathology. Both innate and adaptive immune responses seem to occur, but the response to different immunotherapies has been mixed. And in particular, we've seen attempts to treat extreme disease with blocking IL-6, for example, blocking other inflammatory pathways, or treating with anti-inflammatories like dexamethasone, all of which seem to have some benefit in a subset of patients, but none of which really seem to be broadly able to reverse the disease state and help all patients who are hospitalized. There's clear evidence of T-cell activation. I'm going to show you a little bit of that, suggesting that there's an over-aggressive T-cell response in at least some patients, but there's also evidence of T-cell exhaustion, or at least hypofunction of T-cells from other studies. So the question is, which is it? Or do we have overactive T-cells, or do we have a failure to properly engage adaptive immunity? We also have very little understanding of the temporal kinetics of both the innate and adaptive immune response, and we're starting to see some increased insights here, but I'm going to show you a little bit about what we know about the temporal relationship of adaptive immunity to clinical presentation. We also know that there's lymphopenia. Lymphopenia during acute viral infections is not terribly unusual. What is unusual here, and I'm going to show you the data in COVID-19, is the duration of this lymphopenia and the fact that it's very T-cell biased, and I'll talk a little bit about that. And the consequences of that lymphopenia for some of the other immune phenotypes are really poorly understood. We also know that these patients make antibodies. In fact, many of our hospitalized patients show up on the first day of hospitalization with antibodies. This coincides with a very robust plasmablast response in some patients. So despite this activated B-cell response and antibody, patients are still sick, and convalescent plasma that is adding more antibody to these patients can sometimes show promise. So one of the key questions when we started out with this work in late March or early April was really to understand what is a typical immune response to SARS-CoV-2 in these hospitalized COVID-19 patients. And so to do that, we set up a biobanking protocol and really an infrastructure at Penn to take advantage of the hospitalized patients and try to really learn from them in real time and return some information back to the clinic, hopefully to improve clinical care. This was built really on a protocol that Nuala Meyer had set up in critical care to study sepsis and ARDS pre-COVID. She's amazing in her clinical work and really positioned herself to pivot directly to COVID-19 in the third week of March. And by the end of March, we had already started to acquire samples together with Mike Betts, who really helped pioneer some of the profiling side of this, and fed those samples into a series of biobanks and processing units to make sure that we had the ability to study this blood in real time, and then to apply a series of deep immune profiling platforms to understand the diversity and heterogeneity of the immune response. And I'm going to talk only about a few of these. All of the patients had extensive serological analysis by Scott Hensley's lab. I'm going to show you mostly data for high dimensional cytometry. These are 25 to 28 color fluorescence-based flow cytometry panels, although there's some CyTOF data as well that I'll comment on. And then a bunch of other analyses going on that I won't mention too much about, including analysis of serum cytokines and whole blood and single-cell RNA-seq. Again, this was a collaboration with Mike Betts' lab. I'm going to talk to you mainly about the data generated from our lab. Data from Mike's lab that was also published recently looked more at the innate immune system and perturbations in the innate immune system. But overall, over about 8 weeks, we were able to recruit 140 or more COVID-19 patients, most of whom were in the ICU, although in the latter parts of recruitment, we got some non-ICU patients. We also recruited about 40 patients, 40 people who had recovered from COVID-19 and got blood from them somewhere between 2 weeks to 6 weeks post self-reported symptom resolution. Now, these patients are a little different. They were not hospitalized, so the severity of their disease was certainly different than the ICU patients. And then we have about 70 healthy controls to whom we can compare. In the last few minutes, I'll touch on a pediatric cohort that we've developed. Laura Vella has really spearheaded this in our group with about 30 pediatric COVID patients. This is one of the larger cohorts that we're aware of, including 14 of those patients with the multi-inflammatory syndrome in children, or MIS-C phenotype, which is a really distinct phenotype that I'll mention a little bit later. And then we're also studying cancer patients who get COVID and understanding the relationship between cancer as a background change in immune status, cancer and its treatment, and the manifestation of SARS-CoV-2 infection. So let me just walk you through a little bit about what the cohort looks like. So I'll use this sort of color scheme throughout the talk, healthy donors in green, these recovered donors in blue, and the COVID patients in red. And again, we have a total of about 150 COVID patients in the cohort. 125 of those we have deep flow cytometry data for, and you can see the similar breakdown for the other two cohorts. We are a little bit age skewed, as is typical. The COVID patients are a little bit older, with an age range really from 22 to 90, but a median of 60. A little bit older than our control groups, and so keep that in mind. Pretty even gender balance here for all the cohorts, actually. And they display with the typical clinical symptoms, high ferritin, D-dimer, CRP, a relatively normal white blood cell count, but lymphopenia, as I mentioned. The lymphopenia occurs without really a change in other cell types. In fact, there's an increase in granulocytes, and the paper from Mike Bett's lab shows really a marked increase in neutrophils in essentially all COVID-19 patients. On the bottom right, what you see is a series of clinical features that were extracted from the clinical metadata, and the point is not really to identify any individual correlations in this matrix of correlations between these clinical features, but to point out two things. One, that it actually took a major lift for those of you that do this kind of work to extract about 50 features of clinical metadata, annotate it, make sure it's valid, double check it all, and be able to put it in a format that we could use it to compare to the immune features that I'm going to show you. So this is really a high dimensional set of clinical data that really is not always present with a lot of these clinical kinds of studies. The second thing is that you do see patterns here, and I'll talk about these patterns in more detail as we go through, where there are relationships between, for example, different measures of disease severity, and that's what this big red blotch in the middle is. But embedded within that, you'll see things like whether or not the patient received vasoactive medication or something called a PACHI score. And so both of these are indicators of organ damage, vascular problems, and organ failure, of course, correlating with other metrics of disease severity. So what we wanted to do was really understand the profiles of the immune cells in these patients and how they relate to some of these clinical features. Again, to do that, we built a 28 color or 25 to 28 color flow cytometric panel, depending on where we were in the pandemic. And we were able to look with high dimensionality at B and T cells and other leukocytes and some of the other approaches that I'm not showing you. And the one thing we see very clearly early on is, in fact, patients are lymphopenic, but this is a T cell biased lymphopenia. You can see that here in the T cells. B cells numerically, or at least in frequency, are not affected. In fact, there's a proportional increase in the non-B, non-T cells in this gate. We think this is a proportional increase, not an absolute increase based on the actual cell counts we get from the clinic. But the other thing you'll notice from this is it's affecting CD8 T cells more than CD4 T cells on average. It appears that there's more severe loss of CD8 T cells. And in fact, you can quantify that by looking at changes in the CD4 to CD8 ratio in different ways, relating it back to the clinical counts. So what we have here is a lymphopenia in this viral infection, but the lymphopenia is very biased to CD8 T cells, and in fact, it's actually quite prolonged. So in other respiratory viral infections, you do see a transient lymphopenia. It's usually occurring around the time of clinical symptom presentation, but resolves very quickly within a couple of days. It's also not typically biased to CD8 T cells. The only other setting where that's been reported that I'm aware of is actually experimental Ebola infection in primates. If you look at those T cells that are present in a little bit more detail, what you find using activation markers like the combination of CD38 and HLA-DR or expression of Ki67, in other settings, you can use those markers to identify antigen-specific cells responding to acute viral infection or live attenuated vaccines, or in fact, cancer immunotherapy, where the tumor-specific T cells show up in either this upper right quadrant or this Ki67 positive gate. What you see in healthy controls and actually these recovered donors, that there's a low level of T cell activation in the steady state. In the COVID patients, you see quite a range. You actually see many patients have this very robust T cell activation, and noting for Ki67, this can be more than half of the CD8 T cells in the blood actually have expression of Ki67, indicating recent activation of some sort. But you also see other patients that actually have very low levels of activation, in fact, not different from the healthy controls or recovered donors. You can look at this in a little more detail, taking advantage of the high dimensional data. And as you display this in sort of in t-SNE space, what you see is that the healthy donor and recovered donors are quite similar. They're not identical, and we could talk about that later if anyone's interested. But the COVID-19 patients really display sort of striking reorganization of the phenotypic space of their CD8 T cells. You can go in and sort of overlay this and ask what's happening, and this sort of middle channel of this t-SNE plot is actually where a lot of the Ki67 and CD38 is. This is your sort of activated proliferating cells down here. You also see this spot here in the recovered donors and the healthy donors that's missing from the COVID-19 patients on the CD8s. This is in fact where your TBET positive and CX3CR1 positive effector-like CD8 T cells are located. And not in every patient, as I'll show you in a minute, but in most patients, that population is largely missing for some reason. You can identify clusters within this data and then ask how those clusters are related to COVID-19 versus healthy controls. And what you find is that clusters that are overrepresented in COVID-19 include clusters that have these activated proliferating cells. So this is cluster 14 here, illustrated down here. And other clusters that are depleted, like this cluster 11 that contains these effector-like cells. Although you'll see there are some COVID-19 patients that have normal, if not maybe even elevated representation of that cluster. And for the T-cell geeks in the audience, you can sort of display this overlaying the background gray of a healthy control and see in some cases really profound T-cell activation here in this cluster 14 with PD-1 and Ki67 being co-expressed. We interpret that here as being T-cell activation, not necessarily T-cell exhaustion, though if you use other markers that are a little more specific for T-cell exhaustion, you might convince yourself that at least in a subset of patients, there may be some level of T-cell exhaustion occurring. You can also do this for CD4 T cells and you get the same picture in terms of overall T-cell activation. Many patients have very increased percentage of activated CD4 T cells, although below the green bar, you'll notice there are some patients that really don't seem to have activation of the CD4s. Overall, there's increased proliferation, PD-1 is up, you see a change in the TFH signature, and unlike the CD8s, you actually see a major increase in the effector-like CD4 signature. And that's illustrated here, again, projecting this in t-SNE space. You can see this cluster down here on the lower right, which actually overlaps very strongly with expression of TBET and a little bit of PD-1 there, although it's this bottom part that really has a lot of TBET. There's another little knob on the right, which has actually the highest expression of TBET that's not separated out very clearly, that's actually different in COVID-19 patients. But again, sort of pulling out the individual clusters, what you find is we have an increase in COVID-19 in cluster 14 and 15, which are these clusters that contain the activated proliferating CD4s and also this TBET CX3CR1 cluster of effector-like CD4 T cells. So if you go back and actually then ask how this all relates to clinical presentation, basically looking at correlations between the immune features and the clinical features, what you find, there are a lot of things you can find with this kind of analysis. I'm indicating the p-values here with asterisks and then adjusting for an FDR of 0.05. It's really the ones with boxes around them that have the highest confidence. So a lot of the other data here probably is meaningful. What you find is that the really activated CD4 T cells correlate with a measure of multi-organ dysfunction, so Apache score, correlate with high ferritin, correlate with procalcitonin, and negatively correlate with lymphocyte counts. A lot you can extract from this. So the number or frequency of naive T cells negatively correlates with some measures of disease, including procalcitonin levels. Activation of the C4s positively correlates with ferritin and also other measures of disease, Apache score, procalcitonin, and others. And negatively correlates with lymphocyte counts, suggesting perhaps a role for lymphopenia-induced proliferation as well as just T cell activation. Many other relationships are embedded here that we could talk about later if people are interested. There's also massive activation of the B cell compartment, and this was really one of the more striking observations when you look in the COVID patients. So typically in healthy controls, and I'm not showing you a flow plot, but the data is here on the graph, you really see very few plasmablasts in the peripheral blood, usually less than 0.5% of your circulating B cells. In many of the COVID patients, you could have 30, 40% of your circulating B cells are plasmablasts. In fact, our record at this point, a patient that Mike Betts' lab looked at had close to 90% of the circulating B cells were actually plasmablasts. And no, it was not a leukemia or a plasmacytoma. So this was really striking. And to put this in context, when you look in acute influenza infection, you see somewhere in the range of 5 to 15% of the circulating B cells are plasmablasts. In acute Ebola or acute dengue hemorrhagic fever, you might get to 30 to 40, maybe 50%. So what we're seeing here in COVID-19 for at least some of the patients is on par with very severe acute viral infections in humans, including Ebola and dengue. Now, what's interesting about that is the plasmablast frequency actually did not correlate with the level of circulating virus-specific or spike-specific IgG. It also did not correlate with the frequency of activated circulating TFH-like cells in the blood, suggesting that perhaps the plasmablasts are making antibody to other determinants of the virus that we're not measuring here, or the possibility that there might be bystander activation of plasmablasts in this case. What is interesting is that the circulating activated TFH correlated reasonably well with the level of spike-specific IgM and also IgG, suggesting that at least some of the antibody response is coordinated through events that likely involve germinal center biology. I say that because we've previously shown that these activated TFH in the blood do actually reflect in some way activity in the germinal center lymphoid tissue. Okay. So how are these responses connected to disease severity? So we took advantage of the fact that all the patients have this measurement of disease severity with this NIH ordinal scale that I think actually has now been reversed in terms of the way the numbers are used. Nevertheless, 1 here is most severe, being death. Hospitalized patients range from 2 to 5 or 2 to 6. Non-hospitalized patients are 7 and 8. So we only have from a range of 2 to 6 here. But what you see actually is the lymphopenia correlates very well with disease severity. And you start to see some hints about relationships between T cell activation and disease severity. But the thing that we noticed from this data was actually less so about the few statistically significant relationships that we could see, and more actually about just the display of the data, just the enormous range of activation phenotype that you might see, including patients that have lower disease severity and yet extremely high T cell activation and vice versa. So what we decided to do was actually extract individual immune features from this high dimensional cytometry and relate them to donor type across the top. Again, red being the COVID patients, green the healthy controls, and blue the recovered donors. And then on top of that, layer on disease severity. And in fact, in many of these patients, we actually have multiple time points for disease severity. What I'm projecting down here is the day 0 immune phenotype, asking whether the immune phenotype at presentation predicts the trajectory of disease. And now what you might be able to convince yourself here is that you start to see patterns coalescing, or at least groups that segregate out in this sort of k-means clustering. And then in fact, you might say that there's a group here in the middle, group 2, where you see more progression to severe disease and mortality. There might be an enrichment here of activated CD4 and CD8 T cells, perhaps with some activated TFH. Another group over here, group 1, that also has some mortality, but actually seems to have a relative paucity of that T cell activation, maybe more plasmablasts and so on. And so you could do this a number of different ways. If you only looked at B cells, you actually get a different pattern. And maybe there are 3 subgroups here. And so you could do this whether you focus on CD4 T cell activation, B cell activation, plasmablasts or B cells, and CD8s. And what occurred to us as we were looking at this is this was highly subjective, that the things we were putting on the y-axis were things that we just thought might be important. We figured we needed a relatively unbiased approach to try to integrate all of the features from the immune profiling. And so at the risk of being a little bit pedantic, let me just sort of give the explanation of at least the way I interpret what UMAP allows you to do. So UMAP is just another high dimensional data visualization approach, uniform manifold approximation and projection. And what's nice about this is it allows you to take very high dimensional data, as illustrated on the right here, and just project it in 2-dimensional space, preserving the relationships between the different features. It's nonlinear dimensionality reduction. It allows a low dimensional representation of the structure in high dimensional space. It's similar to TSNE, which I was showing you earlier, but one of the things, there are actually 2 things we really like about it. It preserves more of the local structure in the data, which we thought was very important here for things that are closely related in this kind of flow cytometric data, different from other data types. And it is massively faster to run with a data set as large as what we had here. So what we did is we took the 27 parameter flow cytometry data, which gave us about 200 discrete features that we could extract easily, and we used that to build the UMAP. There are about 550 discrete immune features when you take all the pairwise and triple combinations of things and so on in the 27 parameter flow cytometry data. And we projected that along with the 50 different clinical parameters onto this UMAP. When you do that, you get a picture that looks something like this. So you get a picture where you can resolve all of the immune diversity into two components, component 1 and component 2, oriented here roughly on an X and Y axis. You can then overlay on that disease severity, here in sort of darker is more severe, mortality data in the triangles. And what you'll see is you also separate very clearly the healthy donors and recovered donors from the COVID patients. The component 1 axis correlated actually very strongly with disease severity, so more severe disease further on the right here. And you could actually then project on top of this and ask about different immune features. So for example, if you look just for activated CD4s by HLA-DR and CD38, what you find is they show up in this upper right quadrant, meaning that that immune feature actually contributes to both component 1 and component 2. You can instead take effector-like CD8 T cells, TBET positive CD8 T cells in this case, and that immune component really, that immune feature really contributes almost entirely to the directionality of component 2. Similarly, plasmablasts contribute more to component 1 than component 2, and you can go on through this, through all the iterations of individual features if you want. But you can resolve that even more, and so to sort of distill out some of that information, if we look at some of those clusters or discrete types of B and T cells that I illustrated earlier on and ask how they relate to component 1 and component 2, what you find is, just as examples, the TBET positive, CX3CR1 positive effector-like CD8 T cells really correlated much more strongly with this component 2, whereas the activated CD4s really correlated very strongly with component 1, although you'll see that the effector-like CD4s correlated a little bit more strongly with component 2. And similarly, you can go across the board. The TFH are largely negatively correlated with component 1, that's these clusters 2 and 3, whereas plasmablasts are positively correlated with component 1. You can also ask about the relationship to clinical features, and in fact, component 1 correlates very strongly with many of the features of clinical disease, including this Apache score, NIH disease severity score, and other features of disease. Whereas this component 2, surprisingly to us, actually did not correlate, obviously, with any clinically measured components of disease. So immunotype 1, we're calling it, because component 1, immunotype 1, is highly activated CD4 and CD8 T cells, altered and really, frankly, missing the normal features of TFH and high plasmablasts. And this correlates with disease severity. The immunotype 2 actually correlates best with these effector-like CD4 and CD8 T cells, memory B cells, and proliferating memory B cells in particular. So you can then ask a series of other questions about the trajectory of patients in this overall UMAP space. So how does that change over time? And so you can take sort of the top quartile of patients that have the biggest movement in their immune features and the bottom quartile that actually don't move and over 7 days in the hospital stay exactly where they were in their immune phenotype, and ask how the change in those immunotypes actually correlates with clinical disease. And there are so many interesting things here. I don't have time to really go into all of them. What you find is when this component 1, containing a lot of the activated T cells and other things, when that goes down, when that changes from day 7 to 0, that is correlates with coagulation complications, disease severity, and actually, when it goes up over time, you actually see an increase in antibodies, so an increase in IgM from day 0 to day 7. So that means that as you're getting more T cell activation, you're also getting more antibody, especially IgM suggests that the increase in these immune features might be giving us a pseudo-temporal picture of the immune response just kicking into gear. Whereas when this goes down, this might actually reflect the immune system on the back end of sort of the peak of an acute immune response. You can do the same thing with component 2, and actually the relationships here are completely different. Surprisingly, component 2 seems to have some correlations with actually ABO blood type. We don't understand this at all at this point, but might suggest some relationships to some of the features that have been previously associated with blood type relationships in COVID-19. The other thing we see is that there's a relationship here with CRP that may be relating to some ways that this component 2 is capturing immune responses. And then bigger changes over the first week seem to correlate with changes in antibody most strongly. So the more dynamic your immune response is on the T cell and cellular side, the more likely it is that you're having changes in antibody as well. Now, let me tell you about just two last things in the last couple of slides here. The first is that you may remember, as I was going through some of the early slides, there are clearly patients who don't display this sort of immune activation phenotype that I've been talking about. In fact, fall within the threshold of what we see in normal healthy controls and recovered donors. If you map where these patients fall on this UMAP projection, they fall largely in this area, sort of the bottom middle. And so what that means is they essentially have a very low score for component 1, and in fact, a negative score for component 2. What's interesting about all of this is, so you can see that they negatively correlate with the immune components, they negatively correlate, their immune features negatively correlate with disease severity, but yet we still see mortality. And more broadly, what you notice on this is we see mortality in all locations on this UMAP projection, suggesting that the reason that patients succumb to this disease may be diverse. There may be very clearly immune-contributed pathology over here on the right, and there may be other ways that patients succumb that actually seem to negatively correlate with immune activation. All right, just in the last two or three minutes here, I want to transition to currently unpublished data to ask what happens in kids. Now, we all know that kids with COVID-19 actually seem to do better. The mortality rate in patients under 18 is actually incredibly low compared to adults, still not insignificant. But what's most scary about COVID-19 in kids is actually this presentation for what's called multi-inflammatory syndrome in kids, MIS-C. These patients display or show up clinically with sort of very severe vascular complications that are quite acute. The vast majority of times, they actually recover and do quite well. But we wanted to ask about the potential immune features of pediatric COVID and MIS-C infection. And so we used roughly the same structure here, and I'm just going to hit the highlights because of time. We have 30 patients, roughly half of whom are these MIS-C kids. They tend to be slightly older. You'll notice while the age range for some of these overlaps, really it's the pediatric COVID where we have the youngest kids, and really we only see MIS-C in kids who are older than 5. Typical clinical presentation, lymphocyte counts are low, ferritin is high, nothing surprising there, and just a minor note, we're separating the non-MIS-C kids into those who present with ARDS and those who do not. These kids are lymphopenic like the adults. You see proportionally fewer T cells, and this is more so in the MIS-C kids, whereas the pediatric COVID actually tend to be not quite as lymphopenic as the MIS-C kids. But both the pediatric COVID and MIS-C seem to have less CD8 T cells. Now the comparison here is actually healthy adults. That is a caveat. We just couldn't recruit healthy pediatric samples during the pandemic. They do have, the MIS-C kids especially, have adult-like activation of CD8 T cells, where the pediatric COVID seem to have less CD8 T cell activation. And you'll notice the pediatric COVID are really on the higher side of the adult spectrum of T cell activation. You can correlate these different T cell features with clinical features as we did in adults. The numbers are a lot lower, so our p-values and statistics suffer from that. But the general associations of T cell activation in pediatric COVID are consistent with adults, that activation of T cells correlates with measures of disease severity, or at least is associated with measures of disease severity. But the picture in the MIS-C kids is actually quite different. Just the relationships do not appear to be the same. And you can do sort of a computational subtraction of these two to basically ask how different are these correlations for MIS-C versus pediatric COVID. And what you find is that they're strikingly different. The relationship between the immune response and clinical presentation of disease in MIS-C is different from pediatric COVID. Now we did a lot of the T cell phenotyping and all the things that I talked about in adults. And I'm just, I want to talk for the next minute or two just about one feature that came out of this. and that is the identification of the CX3CR1, CX3CL1 axis as being highly interesting in MIS-C, and I think actually telling us something about the overall phenotypes in vascular complications in COVID. So this axis has a role in both myeloid cell and cytotoxic lymphocytes, plays a role in microglial cells, but also homing and migration of cytotoxic lymphocytes, especially CD8 T cells and NK cells. CX3CR1 positive CD8 T cells are vascular patrolling cells that recirculate through the vasculature, and they are almost always more cytotoxic and usually encompassed within the terminally differentiated subset of CD8 T cells, the TEMRA in humans or the KLRG1 high cells in mice. There's also a clear role for this axis in cardiovascular disease and atherosclerosis that I'm happy to talk about at the end, but suffice it to say this was one reason we decided to look at these cells in more detail. Now, at first glance, nothing that surprising. So this is just CX3CR1 staining in a healthy adult. And in fact, the frequency of these cells across even the adult severities of disease and pediatric patients was not that remarkable. What was remarkable, however, was the activation of these CX3CR1 positive cells. Using HLA-DR and CD38, MIS-C had more T cell activation than non-MIS-C, which I showed you earlier. But in fact, the activation of these CX3CR1 positive cells was remarkably elevated in MIS-C compared to the non-MIS-C kids. And that's graphed over here, where you can see that even compared to the adults, the MIS-C kids had remarkable activation and/or proliferation of these CX3CR1 positive cells. So what we then asked was whether that immune phenotype had any relationship to the clinical presentation of disease. And in fact, if you just ask whether patients received vasoactive medication and how that related to the activation of this CX3CR1 positive subset, there's a very strong correlation or relationship between the presence, the frequency of activated CX3CR1 positive cells and whether or not those patients received vasoactive medications. So we wanted to then ask whether this pediatric phenotype gave us any insights into what might be happening in adults. And so we did basically the same analysis in adults. We had to use a different readout, and that's basically clot complication. We found that actually when, if a patient had a clot complication, they were more likely to have elevated activation of their CX3CR1 positive CD8 T cells. So I'm going to go through this quickly. We could project this pediatric data onto the same kind of UMAP. We could look at the trajectory of those patients over time. And one of the things that was apparent in the MIS-C kids was that over the week in the hospital, there was massive reduction in the overall immune activation. So this is just draw 1 and draw 2 in a series of MIS-C kids where their overall CD8 T cells are dramatically activated at presentation, and then it resolves incredibly quickly. And you can see that here just in the trajectory of change for a bunch of markers of T cell activation in the MIS-C versus the pediatric COVID. Now, looking at the time frame, it's really to the left of this dotted line that you want to compare, where a lot of the pediatric COVID really have sustained or not changed activation features during that same time frame where we can measure the MIS-C kids. And then looking at the change in the CX3CR1 positive cells, indeed you see the same thing, that actually you see a reduction in the activation of these cells in the patients we have longitudinal data on for MIS-C, whereas that's not so apparent in the pediatric COVID, where you see basically sustained or even increasing activation of those cells over time. The reduction in activation correlates with improvement of many clinical features in the MIS-C kids, return to almost normal lymphocyte counts, platelets seem to return to more normal range, decrease in CRP, decrease in D-dimer, procalcitonin, and so on. So these MIS-C kids present with this unusual immune activation phenotype, and then it resolves quickly. So just to summarize, and then hopefully I'll have enough time for questions, we see tremendous immune and clinical heterogeneity in COVID-19. There's CD8 biased lymphopenia that may relate to some of the CD8 T cell activation phenotype. There's a massive plasmablast response, really rivaling what we see in acute Ebola and dengue infections, and surprisingly stable T cell activation and plasmablast responses over time in at least some of the adult patients. I didn't really show you the details of that data. We identified 3 immunotypes that really are linked to different clinical presentations that might allow us to think about ways to subcategorize patients and select treatments that specifically address their immune phenotype. We identified the CX3CR1 positive vascular controlling CD8 T cell in MIS-C kids, and that may give us some insights into the vascular phenotype that those patients present with, and also the vascular complications in adults. And of course, the whole goal here is to link these immune features to potential clinical treatments that might be able to more specifically address the problems in the individual patients. And lastly, but most importantly, this was an enormous team effort. The adult work was really spearheaded by a group of actually 7 co-authors, co-first authors on that study: Divich, Josephine, Amy, Allie, Jen, Cecile, and Derek, who really all equally contributed to making that come together. It was a team effort also with Nuala Meyer on the critical care side, Mike Bett's lab, really partners for all of that work. We had massive help in processing of these samples by what we're calling the COVID processing unit, and a lot of those folks are listed here. Clinical coordination from a big team. And on the pediatric side, this work was really spearheaded by Laura Vella with help from Sarah Hendrickson, and a big team on the CHOP side in Philadelphia with Ed Behrens, Dave Tichy, Caroline, and Hamid Basari. So I'll stop there. Hopefully we have time for questions. Sorry if I went just slightly long on that.
Thank you so much, Dr. Wherry. You got through a lot of data and we're getting some really interesting questions. So I'll dive right in. Let's see. I'm going to go sort of in order based on the last, we're getting a lot more questions on, I think, your last few slides. Let's see. So were you able to get BAL from any of the infected subjects to determine or compare T-cell responses in lung versus blood?
No, it's a great question, and of course something that's really critical that the field tackles. Early on in the pandemic, for safety reasons, we weren't really able to do that on a systematic basis. So we don't have that comparison here. What we do have, at least on an anecdotal basis, is we did get rapid autopsies on about a dozen or maybe close to 20 patients now. And at least in a handful of those, we have paired blood from when those patients were in the hospital. That work is still very much in progress. It's going to be anecdotal because it's probably only 2 or 3 patients where we'll have that paired information.
Sure, yeah, great. Okay, is there evidence of plasmablast response with neurological degeneration? Recently reported, it seems like the ACE2 responses in neurons. Do you find evidence of that with the plasmablast response?
Yeah, we don't have any data on relationship to neurological complications. That's actually not something in the clinical data. It highlights, though, the need to really track these patients after recovery, quote, recovery or release from the hospital. And many of us are now, many institutions are now building these post-COVID clinics to try to address some of those questions.
Mm-hmm. That's great. I imagine that's a huge effort, but probably really worthwhile to do and to track and follow these people over time.
Absolutely. It's going to take a big lift, but we're very excited to do that.
Great. So related to the cohort, I had a question on if you have profiles for obesity or diabetes or other disease indicators in your COVID cohort.
Yeah, absolutely. All of that data is there. And it's interesting, if you really dive into some of those bigger correlation graphs, mm-hmm. you will see relationships of pre-existing cardiovascular disease, obesity, and diabetes. Our cohort, and this is interesting and maybe something to take advantage of combining these data with other data, our data is highly enriched for these comorbidities. It's a pretty heavily African-American cohort, has a lot of bias towards obese and other previous or pre-existing comorbidities. Some of those separate out with relationships to the immune features, but not as strongly as some of the other clinical features.
Okay, so we had a question on the flow data. If you mentioned combining signals from the 28 color flow data to define features for dimensionality reduction, could you please elaborate on how you did that?
Yeah, I wish I could. I mean, I can, but there are a lot of computational details here. So this actually was one of the reasons why there are 7 co-first authors, because the pipeline to get this done properly really involves incredibly detailed flow cytometric quality control, manual gating, and really adjustment to make sure every compensation is 100% perfect. Otherwise, that's what will come through a lot of your computational analyses. The individual gates were then set in stone and moved forward. That discrete information was then fed into a series of computational pipelines in R, where we could use those to actually contribute their correlations with each other, which drive the UMAP projection and the t-SNE projections. So we used a series of computational approaches that are all outlined in the paper that probably, not only can I not do them justice, but we probably don't have time to go into all the details of how.
Sure, sure. We can share that paper though. I know it's
Absolutely.
available, I think, in our newsletter, the COVID report, but we can post it when we share the video recording
That's great.
later on. So related to the UMAP trajectory analysis, we had a question: if you combine the adult and the kids data with the UMAP trajectory analysis, would you see a marked trajectory potentially connecting the children and the adult phenotypes?
Yeah, it's actually interesting. So one reason that we use the UMAP projection is it's very resilient to adding new data. And we did build a new UMAP that incorporated all of the pediatric data. And we did that because we actually had to gate slightly differently, even the adult data that we pulled into that pediatric analysis. And in fact, it looks remarkably similar to the original UMAP. So you can actually then map those pediatric immune phenotypes onto the adult immune scaffold. And what was initially surprising to me, and actually I showed it very quickly, the pediatric cases actually scatter across the adult map just the way the adults scatter. What you can see there is there are a few of those pediatric COVID patients that actually map to the healthy controls. And it's a little bit more proportionally than we saw in the adults. That may indicate either the very young patients that haven't mounted an immune response, or the fact that having a little bit more of a quiet immune response may actually correlate with less severe disease pathology if there's an immune pathology arm in some patients.
It's an interesting suggestion, yeah. So we talked a lot about the kids in the cohort, and I think earlier you showed a graph where some of your older cohort, did they go up to age 90? Could you comment on the cohort and just the implications for this type of work in older adults, so like over 65 or 75?
Yeah, I think our oldest patient was 90 years old. Certainly age is a cofactor here. We know that. But of course, we all also know is that this is a really unusual infection. So almost all viral infections, and actually many other infections, have this sort of U-shaped mortality curve, or at least U-shaped morbidity curve. And this one is not. The young kids really do not have the same pathology as the elderly. We've been talking about it, and there have been a number of suggestions of the potential to use the immune signatures of COVID to reveal underlying differences in immune health, and why we're seeing this unique display of an elderly associated pathology, but not the very young pathology is quite interesting. We don't have any great insights into that. It may have to do with this sort of inflammatory phenotype and the fact that as you age, you accumulate more effector-like T cells. The corollary of that means that for that to manifest with disease pathogenesis, there has to be some bystander activation of those cells, because they can't possibly be SARS-CoV-2 specific. So we don't understand this. It raises a lot of interesting hypotheses that are, eminently testable here as we go forward.
Yeah, definitely. Are there differences in immune activation in the long hauler COVID-19 patients?
We don't know. It's a great question. We don't have a long hauler cohort at this point. We have anecdotal evidence that that those patients who are hospitalized for 3 to 4 weeks have sustained activation of their T and B cells, but no definitive data yet.
Okay. And is there any indication that the immune systems of pediatric COVID-19 patients show signatures of aged populations, so immune senescence, et cetera, or do they tend to have more evidence for enhanced inflammation, like obesity or metabolic disease? I think you mentioned cardiovascular disease associations before as well. Yeah,
great question. And it's the reason we were so interested in the MIS-C population. And so, one of the initial thoughts when we first started getting that data is that actually the MIS-C kids had a T cell activation profile that was more like adults. So something about their background distribution of non-naive T cell activation was more similar to adults. I'm not sure we would say that it's more similar to the elderly, but they certainly looked less young in terms of the distribution of effector memory, central memory subsets. That really boiled down to what I showed you, which is the CX3CR1 positive subset, which is not elevated in frequency, but definitely is getting disproportionately activated in these kids.
And you may have mentioned this earlier, but do older kids show any comorbidities like type 1 diabetes, obesity, that might lead to MIS-C complications?
Yeah, I think so. We've not mined that data deeply enough, but I think there is some evidence for obesity, diabetes, and some other potential background comorbidities, but we've not mined that in this cohort yet.
Okay, and going back to the immunotypes, do individuals in immunotype 3, the less activated T cell phenotypes have lymphopenia and neutrophilia?
Yes, they do. So we do not understand this immunotype very well. Those patients, essentially every COVID-19 patient, adult COVID-19 patient we've looked at, with maybe one or two exceptions, has the elevated neutrophils at presentation. So that does not separate out this immunotype 3. Some of them do have some elevated inflammation, although we don't have great data on that yet for some technical reasons. But many of those immunotype 3 patients have antibody at presentation. So how that occurs, we don't know, whether that's pre-existing cross-reactive antibody or whether we have these sort of extra follicular antibody responses occurring in those patients. We don't know yet.
So how might these results inform our understanding of immune correlates of protection from severe disease? And which SARS-CoV-2 vaccines in development do you think might most likely be successful? Is there a good understanding of the type of cellular immune responses that may enhance disease in the context of vaccination? These are loaded questions, but at the end of the day, if you could talk about the implications for, vaccine and therapeutic success as well, that would be helpful.
Yeah, I mean, I think, and I'm going to sort of maybe slightly overinterpret our data a little bit, but to sort of give my thoughts on it. One of the ways we've been thinking about this immunotype 1, we're seeing the most strong correlations with clinical disease is that this is a roughly imbalanced response. We're seeing really activated effector T cells. There's a massive plasmablast response. They have some antibody, but maybe the antibody is not good enough or broad enough or something. And so one implication of that is that if we could get a more balanced response where we have good antibody on board ahead of time, we also have a good effector-like T cell response on board, we might avoid this sort of immune activation. So I say that because one of the other things emerging in the literature is this idea that SARS-CoV-2 infection may delay the initiation of the immune response, either evading type 1 interferon for a few days or somehow delaying the initiation of the adaptive immune response. And so if we can, with vaccines, actually jump the gun and have those things on board at the time of infection, we may be able to prevent one of the key things that's always critical for viral pathogenesis. And that is this race before the virus spreads to so many cells or so many tissues. If the virus spreads too widely and then you mount an immune response, you just target those tissues and cause pathology. So having a balanced response on board at the beginning, I think is going to be the key. So I think in terms of which vaccine approaches would be my choice, I think it goes to things that give you both a really good high quality and maybe broadly targeted antibody response together with activation of CD4s and CD8 T cells. I think CD8s are going to be important to have on board here if the antibodies are not completely protective.
Okay, great. So looking more at the immunophenotypes, regarding upstream drivers determining or at least associated with distinct immunophenotypes, especially the first two, can you comment on the plausible contribution of prior antigen exposure to close coronavirus cousins?
Yeah, obviously there's been a lot in the literature on this, and my friend Shane Crowdy and I have talked about this a number of times on the phone. The answer is we don't know. Before I speculate, the answer is we really don't know. We need to identify that those TCRs in these patients to see whether they're coming from cross-reactive or previously primed cells. There's evidence on both sides of the equation here. Now, most evidence would suggest that having pre-existing, even cross-reactive memory CD4 or CD8 T cells might be beneficial. The question is, there are actually two key questions in my mind. One, there are scenarios where if you have memory CD4 T cells without good antibody, you can actually cause pathology rather than resolve infections more quickly. And so perhaps you drive an effector CD4 response and yet the virus is still spreading because not all cells are class 2 positive. And that's the model from one paper that was published that we were involved in years ago. The other side of it is this idea of original antigenic sin. So if you have pre-existing responses and now you get infected, those pre-existing memory cells, whether memory B cells or memory CD4, CD8 T cells, will target the overlapping epitopes more so than generating responses to new epitopes. Now, whether that's protective or not protective really depends on the pathogenesis of the virus and the infection. Certainly on the antibody side, this sort of original antigenic sin is something that we should all be a little bit worried about, given pre-existing antibody to other coronaviruses.
We had a question earlier about the circulating TFH. You showed that anti-spike, and correct me if I'm wrong, but you showed that anti-spike IgG titers did not correlate with plasmablast or circulating TFH. Is that right?
That's correct.
Okay, so, but does the question was, does the neutralizing antibody titers correlate with PB levels or TFH? Do these findings suggest a qualitative defect in the humoral response in COVID patients and not necessarily a quantitative defect?
Yeah, it's a great question. So what our data shows, and I think there's other data in the field on this, the circulating plasmablast response does not correlate with antibody to the RBD domain of spike. Circulating plasmablast response does not correlate with circulating TFH. The circulating TFH do correlate with IgM and IgG to the spike RBD domain. In the assay we use, about 90+% of the time, antibody binding to RBD is also neutralizing. It's the rare exception when it's not. So most of the data I showed you would be neutralizing antibody correlating with circulating TFH. Now the question is a really interesting one because as many of you probably know, there's now evidence that perhaps germinal centers are disrupted in at least severe cases. Data from Shiv Pillai's group and the Harvard Consortium showed that very nicely in at least severe patients at autopsy. There are a couple possibilities for that massive plasmablast response that might suggest distorted or inefficient antibody production. One is, this is all germinal center dependent. We have responses to many determinants of the virus. The spike RBD is just, I don't know what fraction would be, but a very, very small percentage of the proteome of the virus. The second is that you have, so you have large responses to other determinants of the virus. The second is that we're getting some bystander activation of plasmablasts, perhaps driven by inflammation, IL-6, perhaps other inflammatory mediators driving non-cognate plasmablast generation. Third possibility is a lot of this is happening outside the germinal center, and these are memory B cells being pushed to plasmablast responses very, very rapidly. They could be cross-reactive to previous coronaviruses. They could be cross-reactive to other determinants, and because of the distorted lymphoid architecture, we've decreased the threshold for plasmablast activation. We don't know the answer. I think it looks like most of our patients are making pretty good antibody, at least to the RBD domain. So we don't understand yet why many of our hospitalized patients have virus, are very sick, and have neutralizing antibodies. So there's some concern that perhaps once a patient has a robust infection, neutralizing antibody may not be as efficient at preventing cell-to-cell spread or spread of the virus within, there are multiple mechanisms viruses can spread even in the presence of neutralizing antibodies within the body.
And I think this is a related question. Is there long-term damage to the immune system following severe COVID disease? So the capacity for someone to respond to subsequent viral or bacterial infections?
Yeah, it's a great question.
Given what you just described, it seems.
We are worried about this. We have no data. And you could see a little bit of it on the TC plots that I showed. Now our recovered donors, to give a very short answer, our recovered donors were not hospitalized. Some of them were sampled shortly after recovery, 2 to 3 weeks, but they have not fully returned to normal yet in their overall immune architecture. So I think we would not be surprised if there weren't residual features of immune imprint from COVID-19 infection in these patients.
So do you think these T-cell markers, these phenotypes identified, could these be used to predict patients that are going to end up in the ICU? Or is this measured once the patients are already in the ICU? So sort of an after effect?
That's the next stage of this. We have to do that. I think I am optimistic that we could predict patients who are going to progress to severe disease based on at least some of these immune features. Although, that's a prospective observational trial that we need to do.
Absolutely. But it sounds like you may have the capacity and the setup and the cohort, if the resources were there, to maybe follow that over time and to try to make a predictive signature.
Absolutely. I think the infrastructure is here. And as you point out, for all of us, probably everybody on the call, this is a very resource intensive set of operations for all of us tracking COVID. The clinical infrastructure necessary to do this is
Massive.
Non-trivial, yeah.
Yeah, absolutely. Thank you so much, Dr. Wherry. I think we're just about at the end of the time. Did you have any final thoughts for our audience before I just wrap up the day?
Just to thank you and thank everybody for sticking around to the end and listening to our work.
Well, thank you so much. And thank you for our attendees for joining today. We really appreciate your questions and how engaged you are. It makes for a really interesting discussion, which is really the goal of these lab meetings. So thank you for joining.
Thank you very much.
Thanks. I invite you to join us 2 weeks from today on the next Global COVID Lab meeting, which is scheduled on September 24th at 10 a.m. Eastern Daylight Time. Our next speaker will be Dr. Sarah Gilbert from the Jenner Institute at the University of Oxford, who will talk about her work on the development of a vaccine against SARS-CoV-2, starting with the initial plans in January of this year to the initiation of the phase 3 trials just 5 months later. And if you're interested in more research on COVID-19, please sign up for the HVP COVID report. In this newsletter, we present insights from experts all around the globe and highlight the latest scientific articles and data. And lastly, please feel free to visit our website and follow us on LinkedIn, where we're going to upload a recording of this webinar for your review. Stay safe, and we hope you'll join us next week or in 2 weeks on the next Global COVID Lab meeting. Thanks, everybody.